Okay, so we are going to I'm going to be recording chapter 15. We'll see how far I get. I'll probably break it into two, but we'll see how far we get. All right. So screencast, Zoom. All right, so here is a practice question that you should try. All right. This is going to be talking about chi-square analysis, and hopefully we can have time to do this one in class together, but here is one for you to practice. Genetic fun fact of the day, the taste of cilantro is due to a one base pair change called a single nucleotide polymorphism, a single base pair change. So that's a genetic fact. And I'm gonna skip over that. All right, so we are talking about the chromosomal basis of inheritance. And a lot of that is going to be dealing with this fruit fly in a man named, with the last name of Morgan, Thomas Hunt Morgan, sorry. All right. So recall that a human karyotype is all of the chromosomes laid out in kind of an orderly fashion from chromosome number 1 all the way to chromosome number 22. Those are the autosomes, and then we have these sex chromosomes. Notice that we have 2 of every chromosome. Those are the homologous pairs, right? So, and we'll have 2 alleles for every gene, right, on those chromosomes, right? So sex determination in humans is determined by 2 X's for a female and an XY for a male. And this is not the only way you can do sex determination, right? So chickens do it a different way. Yeah, that's the one that's coming to mind right now, right? But because sex chromosomes don't always come matched, right? You don't always have an X and an X. In fact, sometimes you have an X and a Y, and those aren't really homologous in the same way that all the other chromosomes are. So genes that are on the sex chromosomes have unique and distinct patterns of inheritance, all right? And just a heads up on this, the X chromosome is just as big in size as most of our other chromosomes. And it has, many, many, many genes, almost 1,000 genes. The Y chromosome, on the other hand, is very, very small. It only has about 60 to 70 genes. And one important gene is called the SRY gene. This is a master switch. It encodes for a protein that will turn on another protein, which will turn on another protein, which will turn on another protein, all involved in male development. So females that are XX have no SRY and therefore become female. Males have a Y chromosome, therefore they have the SRY gene and they become male. So if a person has 2 X chromosomes, but has a translocation of an SRY gene onto that X chromosome, would they be female or male? Those would turn out to be male, because all you need to be male really is that SRY. I'm gonna pause here. Hold on, stop share. Hold on, how do I pause? Okay, sorry, quick pause. All right, so all you need to be male is the SRY. So if the X chromosome has an SRY, you're gonna be phenotypically male, right? What if you have an X and a Y chromosome, meaning you'd be male potentially, but the SRY was deleted from the Y chromosome? Then you would be phenotypically female. So just some cool facts. All right, so X-linked genes. All right, so when a gene is on the X chromosome, it has a unique pattern of inheritance, and it's said to be X-linked, all right? So oh, sorry, I need to share my screen. I was just talking away in there. Sorry about that. To myself, talking to myself. All right, here we go. So if a gene is on the X chromosome, it is considered to be X-linked, all right? Human colorblindness is an X-linked trait. Here are some fun things to see if you might have some color blindness. This is a really fun test to kind of check to see if you're colorblind or not. So this one, some of you may or may not see it. This shows the letter 3. And this one over here shows the number 5. People with color blindness might not be able to see those. All right, so our little key is fundamentally different when we're dealing with X-linked traits. So for females, for example, we have 3 possibilities for what your alleles would be. You could be, for example, homozygous dominant. You could be a heterozygous, or you could be homozygous recessive. All right, you have to have those X on there if they're X-linked. So you'll always kind of draw it as an X with the allele above it. That's an X-linked trait. The males are fundamentally different, and that's because they don't have 2 X's. They have an XY. So there's only 2 possibilities. You can be dominant for the X-linked trait or recessive for the X-linked trait, right? But not heterozygous. So X-linked human disorders have this interesting thing that they are way more common in men than they are in women, right? And the reason is that they have, for example, oh, my formatting's bad again, darn it. So females have these 3 possibilities. I'm gonna draw them out again. So we have dominant, we have het, we have homozygous recessive. So both of these are unaffected, whereas only the homozygous recessive is affected with the trait, right? So you have 2/3 chance being unaffected. Males, 50-50, right? And you can follow this chart to kind of look to see what is happening. But let's do some of these. So what are the chances that a colorblind male, so colorblind male would have the genotype of the recessive, right? So they're colorblind, so they're gonna have that recessive allele. And having a colorblind daughter, if the mom was a het, so mom is a het. So we're gonna do a Punnett square just like we did before. So we're gonna have the alleles for dad on one side, the alleles for mom on the other, and they're going to come together. So if we're talking about an X-linked gene, we're always going to be looking at 50% female and 50% male. Those are the chances. So the question is, what are the chances of having a colorblind daughter, right? So colorblind daughter would be this one, right? So that is a 25% chance of having a colorblind daughter. What if I ask, what are the chances of a daughter being colorblind? That would be 50%, right? Because you already, if you're saying only daughters, you're getting rid of that, right? So let's do another one. So if a man and a woman produce a colorblind son, so we know what his genotype is, and the dad was colorblind, where did the son get his affected X from, right? So where did the X come from for the son? Right, let's draw out the pedigree and see. So anytime that we're dealing with a male, the Y chromosome is gonna come from dad, right? So this son got the Y from dad. So this little X little b had to have come from mom, right? The Y came from dad, the X had to have come from mom. So mom was either homozygous recessive or a heterozygous. All right. All right, so introduction to, so those are some human examples of X-linked. So now let's look at some flies. Introduction to fly genetics. All right, so similar sex determination in flies, XY male, XX female. So here, males can only get an X chromosome from their mother. So we just kind of went over this in the slide above. So males, because they have to get their Y from dad, their X chromosome comes from mom. Has to come from mom, right? If the gene is X-linked and mom has the recessive phenotype, what's going to happen? All right, so here we're looking at two kinds of flies. We're looking at an eye color phenotype where you can be red, which is the dominant phenotype, or you can have white eyes, which is the recessive phenotype. So the male here had a X big W, and the female had two little W's. So again, I like to draw cursive so that I can tell the difference, right? So if we do the Punnett square, we're gonna put dad's gametes there, mom's gametes here. And what you can see is that the mom over here is giving her recessive allele to all of the males, right? So all of the males are going to be affected, but none of the females. Okay, I'm going to stop there and call this one, we're going to stop here. Alrighty.